We're going to spend a lot of time talking today about some recent research that relates to these new and very popular weight loss drugs, these
Mm,
anti-obesity drugs like Ozempic, and these are GLP-1 drugs. And I want to start out by giving people a sense for some of the basic biology around GLP-1 here. So to
mm.
start, can you just describe what is GLP-1 and what is it doing endogenously in our bodies?
Yeah. So GLP-1 is one out of several hormones that are produced by our gastrointestinal system. So it's produced by a cell type called the L cell in the gut. And typically these hormones, they are peptide hormones, meaning that they are specific size in terms of they have a specific number of amino acids. They are produced in response to ingestion of meals, right? And they are sensitive with respect to macronutrient and even micronutrient distribution. So we know that historically they were discovered for glucose ingestion. You see a secretion of this glucagon-like peptide 1, GLP-1, and it was then identified that this would affect the beta cell of the pancreas to stimulate insulin release. So the first kind of loop discovered in the middle of the 80s was that glucose ingestion elicits GLP-1 release from the gut that signals in the pancreas to potentiate insulin secretion so we can remove the glucose from the plasma. So it's a part of kind of this glucose regulating endocrine system. And so this was referred to as the incretin effect. So that means if you give the same bolus of glucose orally versus intravenously, you would have a much more pronounced insulin response to the oral load because you get the effect from the incretin hormones. And by the incretin hormones are comprised of the GLP-1 and its close sister GIP. So these hormones were identified in the 70s and 80s, and in addition to GLP-1 and GIP, the most well-known gut hormones are peptide YY, PYY, or there's a hormone called neurotensin, CCK, and a few other more well-known peptide hormones secreted from the gut in some type of mixture in response to ingested meals, right? Um, so the endogenous function is typically referred to as this incretin effect to potentiate insulin secretion so we can remove nutrients from the bloodstream and get it deposited into muscle and fat and liver tissue typically.
Hmm. So every time we eat, basically a cocktail of different hormones gets secreted, and they're meant to affect different tissues in the body so that our body can effectively utilize whatever it is we've put into it. So if you eat a meal with one nutrient composition and then a meal with a different nutrient composition, that might necessitate higher or lower levels of glucose uptake or other nutrient absorption. And so when everything's working properly, these gut hormones get secreted in some combination that's suited to enable our bodies to uptake what we've actually ingested.
Exactly. Yeah.
And so GLP-1 has to get secreted, and then it has to activate GLP-1 receptors. Where I'm assuming that these things are located on many different cell types and tissues throughout the body. What are the major tissue systems that respond to GLP-1 that express the receptor?
Yeah. So initially we've been focusing on the endocrine pancreas that have they have these islets of endocrine producing cells that produces insulin, the beta cells, or glucagon, the alpha cell, and they are decorated with these GLP-1 receptors. So that's also why this well-known effect on insulin secretion has been early characterized. Then there are also local neurons sitting in the proximity or vicinity of actually where the peptide is produced in the gut. So, for example, a vagal nerve projecting to the brainstem, probably picking up locally produced GLP-1 signals and transmitting it through a nervous signal directly to the brain. And then this GLP-1 receptor is pretty densely expressed in the hypothalamus of the bottom of the sitting just around the third ventricle of the brain in the bottom of the brain, and also in the brainstem where the vagal nerves is projecting around these two nuclei called the area postrema and the nucleus of the solitary tract. So specifically in the context of obesity and diabetes, it's the GLP-1 receptors expressed in the hypothalamus and in the brainstem we're interested in, and in the pancreatic beta cells for the insulin secretion. And then it's expressed to a lesser extent in other tissues, and we continuously try to kind of discover kind of smaller or more subtle levels of expression on other, for example, immune type of cells, etc., to try and understand what effect the peptide can have on other cell types and systems. But mostly it's expressed in the beta cells of the pancreas and in these two areas of the brain.
I see. So it makes sense that it's expressed in the pancreas, because if GLP-1 is related to food uptake and nutrient acquisition and the insulin response, it makes perfect sense that you would find it in the insulin, which is where in the pancreas where the insulin is secreted. It's also found in the nervous system. So it sounds like you've got the vagus nerve, which reaches down into the gut, so it can sense the GLP-1 there, relay that information back up to the brainstem. You also have these GLP-1 receptors, you said, in the brainstem and the hypothalamus. How are they sensing that GLP-1? Is it crossing the blood-brain barrier and getting into the brain, or is it getting to those areas in some other way?
Yeah, so this is the intriguing part. So these areas are often referred to as kind of circumventricular organs or sites. So they are kind of the just surrounding the third and the fourth ventricular system. The blood-brain barrier is often referred to as being less fenestrated or more leaky. So, and that system is quite advantageous in the sense that this is where the brain is capable of communicating in a neuroendocrine fashion with the rest of the body. So, especially in the hypothalamus, the most lowest region, called the arcuate nucleus, is decorated with receptors for circulating hormones, peptides, and proteins, including insulin, but also leptin produced from the adipocytes. So that area of the brain can actually see peptides and proteins in circulation, and is less protected than the rest of the brain. So it's a kind of a neat design in terms of brain-body communication, right? So the brain can pick up on what's happening in terms of nutrient status and the endocrine kind of factors that signal also nutrient status.
I see. So the most of the brain is protected by the blood-brain barrier. So
Yeah.
there's tight regulation of what goes into the brain. We don't want bacteria getting in. We don't want toxins getting in. We want to control exactly what's going in, because the brain is sort of the master controller of everything. So we've got this blood-brain barrier that's tightly regulating what gets into the brain. It's preventing a lot of things from getting into the brain. But a couple parts of the brain, special pieces, including the hypothalamus, they are in direct contact with the blood. So the hypothalamus is basically directly sampling what's in the bloodstream.
Exactly. Yeah. So if you do labeling studies in rodents, you put a fluorophore or something on a peptide, GLP-1 peptide, you can see that it up concentrates in those regions of the brain, but it doesn't get further into the brain typically.
So for a neuron in the hypothalamus, say, what happens when GLP-1 binds to its receptor? Does the neuron become more excitable, less excitable? What actually happens to neurons when GLP-1 gets released?
Yeah, that's a good question. And at the neuronal level, you see increased excitability, but it is a G protein coupled receptor, so you don't have this kind of immediate depolarization necessarily so it's perhaps more a sensitization of the neuron in those cells. So one of the neurons that expresses the GLP-1 receptor is called the POMC, or the POMC neurons, which is kind of the primary kind of satiating or satiety hormone in the bottom of the hypothalamus. We talk about these yin-yang hormones in a little bit simplified fashion, where the AGRP neurons, and these are because they produce these neuropeptides, express AGRP or POMC. So the AGRP neuron is the hunger promoting neuron, and the POMC neuron is the satiety promoting neuron. And the POMC neuron respond to 1/3 of them expresses the GLP-1 receptor, and will then signal or at least sensitize in response to GLP-1, and will have a little bit be excited, have increased excitability.
I see.
And same goes for some of the neurons in the brainstem.
I see. So the neurons in the hypothalamus and elsewhere that have the GLP-1 receptor, they're sensitive to GLP-1. GLP-1 is basically making them more excitable, which
Mm,
may or may not translate into causing those neurons to fire more directly, but will make them
no.
more sensitive to their inputs. So you've got these two populations in the hypothalamus in one
Mm,
nucleus. You have
mm,
AGRP neurons, which promote hunger, basically, and
mm,
POMC neurons, POMC, which promote
yeah.
satiety, basically. So if the
Mm,
POMC satiety neurons have GLP-1 receptors and GLP-1 goes up, you would
mm,
expect to promote satiety,
yeah.
more satiation, less hunger.
Exactly, and there are also then a lot of indirect communication. So there's a recent work where they're using a method called fiber photometry. So you can actually in the vivo in the live mouse you can read the activity of neurons once they fire. You get like a calcium signal that you can pick up on with an optic sensor, a fiber, and then you can see if you give these analogs GLP-1 hormones that you also can see a suppression of the AGRP neuron. It's not direct because it doesn't have the receptor. So there's a lot of cross communication going
Mm-hmm.
on between these neuronal systems, right? So again, and we can talk about this later. Also, we see also that activity deeper in the brain with some of these type of gut-based hormones, but it's not because we just talked about they don't reach deep into the brain, but they will have feed forward or they will have activate neuronal signals that will then kind of signal further into the brain and kind of promote even affect, for example, the mesolimbic hedonic reward
Mm-hmm.
pathway. But those are not because the peptide is direct signaling there, but it's through feed forward pathways.
I see. So the hormone GLP-1 can promote satiety at the level of the brain by acting on specific neurons in the hypothalamus.
Exactly, yeah, and probably in combination with its effects in the brainstem, right? So there's a study from 2018 where they made a GLP-1 receptor knockout animal where they only ablated or knocked out the GLP-1 receptor in glutamatergic neurons. So they used a marker for glutamatergic neurons called the V-GLUT-CRE mouse, and then they crossed it with a GLP-1 receptor flux mouse. So you only knock out the receptor in neurons that expresses this marker for glutamatergic neurons. And then they tested one of the earlier long-acting versions of the GLP-1 drug, and they saw that in mice that didn't have GLP-1 receptors in the glutamatergic system, the drug didn't work on body weight lowering and food intake lowering. So
Hmm.
based on that study, for example, you can pinpoint that the pharmacological benefits on body weight and food intake is mediated at least through glutamatergic system in the brain. They did the same for the GABAergic system, and they didn't see any loss of effect of the drugs. So those type of combination of these mouse genetic works with the pharmacology can then teach us more about the central modes of action.
And so, because we've got these circumventricular organs that can
Mm.
directly sample the bloodstream of the brain, in parts of that brainstem, the hypothalamus, that means these GLP-1 agonist drugs, the Ozempics and the related drugs, they are actually acting on those areas of the brain directly.
Exactly. And this is something we should probably touch upon now because the endogenous hormone is very, very short-lived, less than
Mm,
2 minutes. So it's debatable whether the endogenous produced GLP-1 ever reaches the brain in a sufficient concentration to signal satiety.
I see.
So we exploit the system with the pharmacology because we can create these super long-lived versions of the hormone that are protected from enzymatic cleavage in the bloodstream. And through lipidation or fatty acid side chain, they also bind to albumin, so they stay in blood circulation for many, many, many days. And in that way, they've been engineered to signal in the brain as opposed to the endogenously produced GLP-1.